I'm Dr. Jennifer Jordan from the VCU College of Engineering in the Department of Biomedical Engineering. And I do research as a biomedical engineer using applications from engineering in the medical field. So that's what biomedical engineering really is. And my specialty is with cardiac imaging. And so I use a imaging technique called magnetic resonance imaging, or MRI, which you might have heard of. And we use MRIs to take pictures of the heart to see if the heart is becoming diseased or abnormal. And so I do a lot of clinical trials working hand in hand with the clinicians. And I'm talking to you from my lab here at the Pauley Heart Center at the medical campus. And so I work very closely with clinicians and apply physics principles and imaging principles to clinical studies. And so you can see on this screen here that we do a lot of fancy image analysis, but we can take some really neat pictures of the heart to not only watch how the heart beats. Here you can see it contracting down, but we also look at how it can have different strain parameters and how it's contracting. So this allows us to look at pictures of healthy hearts and compare them to patients who have all kinds of different cardiovascular disease. And I get to do this in my own research and collaborate with others who do studies all over the country, and we take the pictures and send them here to the lab where we analyze the images and identify what's called a subclinical biomarker. So we know when a heart is failing, but our goal is to find evidence of when the heart is starting to fail, when it's becoming diseased, so that we can help the patient sooner. So we can use our really fancy imaging techniques to do that and identify different tests that we can do for patients. And nothing has to be invasive. We call it a non-invasive biopsy because we can look at the quality of the heart's contraction. We can look at how the tissue changes and if it's got inflammation or scar tissue within it. And so it's a really interesting technique that allows us to characterize in one exam somebody's whole heart health. And we do that non-invasively without any interventions on the body, opening anything up. So it's a really safe and effective approach to cardiovascular care. And we use our imaging principles to do both the analysis and the collection of the images here for our research. So what we have on the screen here is the left ventricle of the heart here in this circle. So that's the main chamber of your heart. That's the muscle of your heart that pumps and squeezes the blood to the rest of your body. And then in this second area over here with the yellow line, that's the right ventricle. So it's partnered right beside the left ventricle. And with our image analysis software, we can contour or draw lines where each of the areas start and end. And we do that in what's called a time series. So we take essentially a movie of the heart beating. So we can watch as it contracts down and gets smaller and bigger. We not only take pictures in one slice, but we take pictures from the top of the heart all the way to the bottom of the heart. So you can see that that cavity or the area here is getting smaller as we contour it down. And then once we've contoured our images, we can create a three-dimensional model of the heart and its chambers here directly from the image. So now we can spin it around and look down into the heart. We can look at it from the side and around and see in 3 dimensions how the heart's structure looks. And if we do this at every time point, we can make what's called a 4-dimensional model and watch how the heart beats in time in this. And this allows physicians to really understand what's going on. Instead of just looking at a single slice in a 2-dimensional flat plane, we can look at it in different areas and see if there's a bulge or if one area is not contracting in with the rest of the heart. So it allows us to do these really interesting techniques here. And then we can look at how things move together in different ways here with our strain analysis too, and see if the heart's contracting synchronously or all together in the same way, or if it contracts in a different manner that would not be healthy. So while our eye may be able to pick that up sometimes, it can be difficult early in the disease. And so with the quantitative techniques that we use here for strain, we can look at that and put a number to what our eye may miss sometimes. Thanks for watching.